Placenta is not mother's organ. Placenta is a baby's organ. Even that is not shared by twins. Each baby has its own placenta. It's like my lunchbox. Baby does not need mother. Baby wants to extract as much as possible energy from mother. So that's why during the pregnancy, baby secrete those hormones to increase the mother's glucose concentration. So there is a pregnancy diabetic effect. 임신성 당뇨병. Because baby wants more glucose to grow, even sacrificing the mother's body. So this is kind of a competition between baby and mother. During the development, the genome sequence in principle, it doesn't change unless the mutation happens. And genome sequence is independent of epigenetic, and this is the same between identical twins in principle. Of course, there may be some small changes, but in principle, genomic information is not changing. cDNA library is the same information for mRNA. So this one contains the genomic, but if you compare, it does not contain all genomic information, partial genomic information. So that's why it's more makes sense. This contains epigenetic information, only express the genes. And even twins different, this does not compress to the histone. Histone compresses to the genomic DNA. Not cDNA library is artificially made in the lab, so it doesn't have a histone. Unipotent, then membrane essential becomes only membrane essential. It cannot be become unipotent. What's the difference with empathipotent actually? What's the difference? Sarah? Pluripotent can become an embryo, and pluripotent cannot? Actually true. Let's compare that later. 조변에게 먼저 따라서.
네.
Okay, other differences are the multipotent as a partial the property. In general, our body has about 25,000 genes. When new life starts, most likely we may need to express all the possible potential gene expressions. So in the beginning, we wanted to express almost everything. So membrane stem cell, most likely comparing to the differentiated cells, they may have, in general, they have a higher gene expression. And knockdown means it's a gene deactivation. And deactivation. knockout means it's removed. That means that this is mutation. So in general, the embryonic stem cells, they have more gene expressions, almost open for everything. To express more genes, you need to demethylate the DNA, so more acetylate histone, and phosphorylation is not clear, histone methylation is not clear. Stem cell has a two phases. One is the cycle union, proliferating and then making exactly same cells. Same cells is a symmetric cell division. Time to time, it makes asymmetric cell division, means keeping one stem cell and the other one is a differentiated cell. So it can be divided into asymmetrically. It cannot form placenta. That's why it is not totipotent. Embryonic stem cell can make only embryo part, but it cannot form the placenta. So how do we are going to talk again later? Each organ has its own mRNA pattern, and so if you make those cDNA, then unique cDNA, similar meaning, unique epigenetic pattern, and mutation is become the count. This is where epigenetic and mRNA expression is. General reaction is that embryonic stem cell is more epigenetically open. Then, differentiated cell, you need to remove those epigenetic markers. And to do that, you need to demethylate the DNA, acetylate the histone, kind of reaction with the general reaction to push those cells to the original and the stem cell reaction. Chromatoma formation is one of them, and chimera formation. What is chimera formation? Suena?
To make conversion. To make a conversion.
Which stage? Okay, let's talk about the later again. This one too. Placenta formation is there. Embryonic stem cell cannot make the placenta itself. So that's why this is not part of the stem cell test. Chromatoma has all of these three. When the embryo is developed, it forms ectodon, endodon, mesodon. Mesodon is the middle. It forms all three. Oma here, Oma means this represents the tumor, right? The tumor, but not malignant tumor. This is benign tumor. How do you distinguish malignant tumor from benign tumor? What's the difference? Jenny? What's the difference of a malignant? How do you define malignant from benign tumor?
I'm not sure.
Jenny? 잠시만 봐. 인생이 transporter, we can change that word invade. If it can invade other tissue and break into, then that's the malignant. So if they keep their boundary, so for the baseline of the membrane, base membrane, then even though they grow, it does not break into different tissue. Then usually we call that benign tumor. So benign tumor is less dangerous than the malignant, but it has a whole different layer. That means if you put stem cell here, then you may see liver cell is growing here, or neuron cell is growing here. Something funny or wrong position that can be found.
I thought that benign meant not harmful, but it's still harmful.
Relatively counts. Right? So if it is becoming bigger and bigger, it occupies the space and the attract more nutrients. Eventually, that may give an extra pressure to other parts. It doesn't kill right away.
But
So
it can still kill you, right?
eventually, maybe, but some people have a very big benign tumor and they live maybe 20 years.
I assume it has to do with the initial location as well.
Yeah, relative terms. So just the medical terms, whether this should be removed right away or it can sustain for some time. So that is the definition. Okay, we can read. We go into it. Okay, I asked you 2 volunteers, but nobody volunteered. Any pressure for exam for next week, maybe? Fine. So today we are going to study about the stem cell and iPS. Okay, stem cell. In the beginning, it forms some kind of a pluripotent, the multipotent cells, and then they divide into different types of cells. And one of you asked the question that, well, ectodon, mesodon, they may have multipotent, but endodon does not. Mesodon, ectodon, they can form different, different. So neuronal stem cell, they can make astrocyte neuron cells and potency. Endodon, actually, it does also have, endodon is usually made to the internal organs. Liver cells, pancreatic cells, pancreatic has alpha cell, beta cell, delta cell, different kinds. So it's not fully drawn here, but actually those organ cells also can be multipotent. So the question is not fully correct. So how do we sort? If the sort means separate or the detect, usually they use specific biomarker for those. So over their biomarker, you can use antibody to recognize those. That part maybe we have a chance to talk about later. Okay, when the egg grows, you have one sperm cell and one egg cells. So egg cells divide in the beginning, especially for human egg cells, fertilized, divide into half. Okay. 8, 16. So 2 by 2 divided. After it reaches about 1,000 cells. And then they move to some part, asymmetrically move like this. And then this part, inner cell mass, this part becomes embryo layer. So when you take this cell out and grow, they become embryonic stem cells. So embryonic stem cell is a part of the embryo part. By the way, outside here, trophoblast, these outside cells, these cells make placenta. So if you just take this out, just grow it, it cannot form the embryo itself. You need to have this linkage of placenta, this part. So altogether, this is coming from the trophoblast here, and then this inner part becomes the embryo layer. So this together you need to make the full embryo development. That's what that property we call totipotent. Toti means total, totally potent. Pluripotent, multipotent in principle, language itself sounds like a similar. Right? Pluri, plural, multi, multis, similar meaning. But in this the field, we define that pluripotent means it can make any part of the cells in embryo. That's a pluripotent. But to be totipotent, you need to help of the placenta to attach the embryo to the mother side. Okay, that's a totipotent. Okay. Now, genomic imprinting. We didn't have a chance to fully discuss about this. So let's just say there is a gene from mother, from father. So this is the same gene, let's say. This gene, interestingly, this gene is one side is methylated, the other side is open. Methylated part is functionally deleted, right? Knocked down. And this side is open. Very interestingly, this gene, female it can make an egg. Sorry, this is male, this is female. Male make sperm cell. Then this gene go to one sperm and this can go to the other sperm. Very interestingly, this gene is both of them is methylated in the stage of sperm making. Funny way, this gene from mother, this gene when it is made into the egg cells, both of those genes are demethylated. And then they will make embryo. And the result is the baby has this gene always active from mother side. Always this gene from father side is deactivated. This is genomic imprinting. This funny situation is discovered during the sperm formation and then the fertilizations. Some genes, father side always methylated. Only mother side open. Those genes usually to protect mother's body. Some genes, opposite way, only father side is demethylated, mother side is always methylated. Those genes usually working only for the baby's advantage, to make the baby size bigger and attract more nutrient from mother side. This is an example of competition between male and female. I gave you the example of the placenta. There's a competition between baby and mother. For baby, the most important thing is himself, the baby itself. So even though mother is sacrificed, I wanted to grow stronger, maybe than my siblings. Then try to attract more energies from mother. Now in this case, main case, I provided the sperm, and my baby is growing. And importance between mother versus baby. Baby is more important to me because it's genetically more identical to me. So they pushes the gene working as baby growing faster and stronger. Mother side, this is my baby. That's important for me, but this may not be the only chance I can have baby. I may need to protect myself for next chance, the next batch of the babies. That's why this kind of gene may be most likely one gene from father side, protection side may be functionally opposite functions. Those have the opposite pattern of imprinting. One is mother is suppressing the babies, to not give full energy to the babies, save to some extent. From the father side, just encourage the babies to grow. That kind of interesting observation is found in genomic imprinting. Those examples are the competition between sexes. So I wanted to hear some examples from you, but anyway, so this is some summary of that. Okay, tetraploid. So before we go to the tetraploid, let's think about here. So let's read this again. Tetraploid, Okay, when we studied the DNA contents, our body is a 2N state. Each one has an N information, so it makes baby, and that's a 2N state. So 2N is our regular, the form of the information, DNA information. So when the embryo is growing at this stage, and if you give a shock here, like a UV irradiation, then somehow they divide, but they cannot, they make a copy of the DNA, but they do not divide. Then one cell may contain the 4N. That's a tetraploid. So 2N state we call diploid. This is the monoploid, but half of this is called the haploid. Haploid is an N, diploid 2, this is normal. Tetraploid, it's not normal. So that's why once it forms a tetraploid, this fast growing part, they cannot survive. They die. But tetraploid outside here, they grow slowly, so they can survive. So that's why this embryo can make a placenta, but it cannot make embryo. Embryo part is all dead. Here, now if you have embryonic stem cell, you inject into this cell. That's the next page. So when they were developing, by giving shock, you kill those the inner cell mass. So this can form only the placenta. Here you take those embryonic stem cells and inject it here. If they can make a baby, that means this embryonic stem cell has a full ability to become organ. Okay. That's the test to see whether this embryonic stem cell is really embryonic stem cell. If they have any defect, then maybe one eye is gone, or the one leg is gone, kind of the defect you may find. If you make a full baby, that means this embryonic stem cell is really pluripotent. So this is the most difficult of the test. If it passes, then this embryonic stem cell is qualified good quality of embryonic stem cell. Okay. That's a tetraploid complementation. Complementation is 상보, mutually help. So placenta part provided and embryo part provided, they help each other and make the baby. This is a tetraploid complementation test. Purpose to check whether the embryonic stem cell is really pluripotent. Okay. Now here, chimera formation is once this cell is growing, now you inject, let's say this is white mouse. You inject the embryonic stem cell of black mouse here. Inject partially. Then they mix it together. Then, and the result of the baby, some part is white, some part is black. That's a chimera. Chimera is different from mutation, and it has actually 2N state, but mosaic structures. Some of those 2N is from white mouse. The other part is from black mouse. It's kind of mixed of different, the origin of those cells. So sounds like this chimera sounds very weird, but actually this is quite common. Let's say sometimes the female has DNA contents as XX, right? One of the X is deactivated, most of the cases. So X deactivation. But if you take the blood and check, sometimes there is XY cells. Can you explain how? After giving birth of boys. So the baby is protected inside the womb, and only connection is the placenta. There's a strong barriers. Usually the interaction of those material is limited, but some of those baby cells can escape and then go into the mother's body, and they can circulate in the body. So this is not very rare. That's why 10 years ago, when they checked the Down syndrome of a baby, usually they put the liver directly into the womb and then took out the liquid, 양수. But it's quite dangerous because it can touch the babies. So these days, they collect the blood from mother's blood system and then check whether those babies, the DNA. So that's possible. That means small amount of exogenous cells are circulating in the body. So that's what they call microchimerism. It's not a very big part, but some cells are from foreign origins. It's also possible. And another case of a microchimerism is one woman is the sterile, cannot generate the babies. But once it gets some blood donation, then time to time they become fertile. Those cells donated from other person, it can activate those production of sperm cells or the egg cells, and it can make the babies. Then you need to check the baby's gene, whether that is donated from mother or from blood donors. That kind of case is also recorded. Okay, so today's lecture is about the stem cell. Here, in the very beginning stages, most likely the cells will open all the gene expressions. And then they move into different directions. One of you asked how they are controlled to go to which direction and which order. Okay, so upper part, down part, how to control those orders. That's another big topic. We are not going to touch that much. But eventually it goes to the some directions. Blood cells, muscle cells, neuron cells, some directions. How do they define it? And it seems like those are chunk by chunk. In the beginning, it's quite often majority of the cells genes are expressed quite often, but chunk by chunk, area by area is closed down. And if you look at those gene expression, so this is gene expression pattern. Yellow color means this is expressed. Blue color is suppressed. Area by area, this is the stem cells. Majority of them is open, all expressed. Some part of the cells, this is the placenta part, and this part is only this part is activated. Give the cell all quenching. And these are activated, any other part are blocked. Block by block. Then this gene maybe you can call placenta genes. This gene may be some specific cell types here. So through moving down from here to each individual of the course, seems like this branch may be blocked, maybe this one, and this branch may be blocked, this one. Chunk by chunk is closed down those areas. Maybe this is the easiest way to control, to make the direction of moving to the liver, or moving to muscle, moving to the brain, kind of situations. So to make originally the pluripotent cells, undifferentiated baby cells, they become adult cells, which has special functions. They don't need all the functions. They just need to keep special function only for the blood, special function only for the neurons. This is the direction of differentiation during development. Okay, that control is made by these epigenetic regulations. So genetic sequence is not changing, but epigenetic markers, DNA methylation and connected to histone acetylation, those are controlled and making all different organs with different pattern of this part this is representing those DNA methylation. DNA methylation here, the black one is methylated, open one usually demethylated, unmethylated. So more open one is more like a embryonic stem cell, more black one depending on which direction. So this process, you can imagine that this is reversible. Methylate, demethylate, you can go back. So if this is a differentiation reaction, in a sense, in the case, this is the DNA, the expression into mRNA. That's why this can be called as a transcriptomics, or it can be epigenomics. Has almost a similar meaning. Let's check this comment first. I can imagine that stem cell therapy can be used for cancer like leukemia. Well, I have leukemia. So that the white blood cells just proliferating and doesn't do much function as immune cells. So kill them by getting the irradiation. And then maybe putting stem cell in there. Well, is that the most efficient way? If I have leukemia, if that is originated from my own gene's function, my gene has some weakness, my gene has some tendency to generate leukemia. And even though I reactivate to restart, still my stem cells can make leukemia again. Isn't it? So in that case, sometimes if I wanted to change my genes, then maybe especially for making these the different cell types, blood cell types, maybe I don't want to use my cells because my cell has a problem genetically. Then maybe better to take somebody else's the bone marrow and then use those cells to replace my blood cells. This is kind of a passive genetic modification. Right? I didn't touch the genetic engineering to change my genes, but I took somebody else's cells, which may be healthier, at least, which does not form the leukemia, and then take it. So this one is, in a sense, maybe quite long history, already has established the history of the cell therapy, the stem cell therapy, adult stem cells, not fully pluripotent stem cell, but bone marrow has a lot of hematopoietic stem cells, multipotent to make every kinds of blood cells here. So there is the part here. But can the stem cell can be used for plastic surgery, not cosmetic surgery? What's wrong with the cosmetic surgery? Cosmetic surgery is very important too. Means it's like making our skins more younger. That's very important, the part. But definitely this one, it's even bigger scale. I lost the leg. Leg maybe rebuilding may take a little more of the efforts to make exact shape too. But if you think about the liver has a problem, liver grows very fast. So if I have the stem cell for making liver, then it can be quite good. You can culture those liver in test tube, my own stem cell, and then my own liver, and then replace it. It's very possible. So that is the promise of, rather than small molecule drugs, utilizing this cell therapy, especially for regenerative cell therapy, utilizing stem cell, is the promise of this field. So embryonic stem cell is one way, or maybe these other stem cells. Representative one is here, the bone marrow, to make the replace with the blood cells. In the video, the clip I showed you, to take the mesenchymal stem cell from lipid part. That also has a lot of the multipotent stem cells. Currently, those cell therapy, main part is those mesenchymal stem cell is the most popular ones. It can form muscle, lipid, and the joint, the structures. So it can be most popular at the moment, stem cell therapy. Now, one of those problems with this stem cell, if you use embryonic stem cell, assuming that we have an embryonic stem cell, it's pluripotent, it can make any forms. It's a powerful, the problem is that it's too powerful. It can form everything. I wanted to have a nose here, but if you have an ear here, then that's a problem. So that's why suppressing those teratomal formation is the one big issue. So partially differentiate to the direction I want to look at, and then stop the retro differentiator going to the different directions. So controlling of proper differentiation is a big issue. Another big issue is if I need to use somebody else's cells. Hematopoietic stem cell also, if you need to find the proper donor, you should check many other immune responses. That's why you cannot choose anybody. It should be relatively close the person to you, maybe your siblings or your uncles, which has a partial connection to the genetic matching. If not, totally third person, independent person may have a strong immune response to each other. So this is kind of limitations to utilize somebody else's and also maybe too premature one. That's why don't you make your own stem cell? Take out your own cell and then make artificial stem cell, that would be quite useful one. Now, that is the starting of this patient-specific stem cell making. In the history, so this person, John Gurdon, he developed this, amphibian, like a frog, and found that by removing the nucleus of the egg cell, by putting the somatic cell's nucleus here, originally this egg cell is an N, right? Haploid. Remove that and then put in those the somatic cell, which has a 2N DNA, putting in here, nuclear replacement. That could make a full body. So that's what he discovered. It's a half a century ago. But that discovery, the people believe that that can happen only to the reptile or frogs, the amphibians. But that has been the proven that this can be done to the mammals too. So the famous Dolly, 1996, this was demonstrated. So for this, so they use 3 sheets. One is egg donor, one is this skin cell donor, and the other one is the womb donor. And then they collected egg cell from here, removed those nucleus, and then from this sheet, put in those nucleus inside here, and then third mother, by putting this into this mother, then A, B, C, whose genetic information Dolly has? A, B, C. Should be A, right? This is the egg donor, and this one is the nucleus donor, and this one is just the womb donor. So this has been demonstrated. People believe that this is almost impossible or very difficult because of epigenetic markers. DNA methylation pattern is different. That's why it would be almost impossible, but they demonstrated that this is possible. Dolly was born. Dolly was not fully healthy, but anyway, this made that breakthrough that even mammals, if Dolly could be done, why not dogs? Why not monkeys? Why not Homo sapiens? In principle, make it possible by doing that. And then that has been applied. This nuclear transplantation method has been tried with humans, published in Science, and big scandal happened in Korea. And here, can you see something wrong? In 2004, the first paper came out that human embryonic stem cell forming is possible using nuclear transplantation. The next year, 11 patients, the patient-specific stem cell was made. It was published in Science again. And can you see something funny in this picture? This cell and this cell. It's the same picture. Just that this is extended a little to this way. Same picture, just extended to this way. Not only this one, there's a multiple the manipulated images were found. So the young scientists, the students and scientists in Greek, they figured out some pictures look quite strange, and then later it was illustrated that this is a total manipulation of those data. None of those stem cells were really made. Maybe the first one, maybe that was true, but none of the 11 cells is really made. So that was a big scandal, big shame. So after that, the major journals, they check these pictures. I said this picture is original, then this picture is expanded this way. So if you submit your paper with the picture data, then any manipulation changed this way or this way, then they reject your paper right away. This is the data manipulated the pictures. That's why you should not touch XY scale changes for any pictures you generate. That's a simple first check you can do, because those every picture has its own pixel. those pixels should be X, Y, scale should be same. If there is any change this way or this way, it will be big trouble. That's why you should be very careful when you make your picture presentations. You should not change this scale at all. Okay? So this was after this the scandal. Now the field of nuclear transplantation, at least for human, is almost black everywhere. First of all, this was the big scam, and second of all, the efficiency was too low. To make one successful nuclear transplantation, you need to use more than hundreds of human eggs. Compared to other species, the human females do not make many egg cells. One person can make maybe 300, 400 for her whole life. To collect these numbers of cells and get only one successful, that's too expensive and too serious a problem, basically. So this approach is totally blocked. After several years, another person, Shinya Yamanaka, he found totally new approaches, quite crazy approaches. He was a medical doctor, not so smart person, he said. And he published one paper with one student. One student worked and then one other professor, they published one paper, only two persons published in himself. The idea was, by looking at those gene expressions, this is ES, embryonic stem cell, and embryonic stem cell blue color is overexpressed one, the green color is the downregulated one. By comparing different cell types, they realized that gene expression is quite different. This gene expression, what do you measure? DNA or RNA? Genetic data, genomic information is not gene expression, right? Then what do you need to measure for this? You need to measure mRNA expression, right? To do that, collect the mRNA, make the cDNA, and then check which genes are overexpressed. So by reading them, and then they focus on these highly expressed genes from embryonic stem cell, and then made those candidates up to 24. 24 candidates which is overexpressed, highly expressed in embryonic stem cell. And believing that if we overexpress 24 of those genes, maybe we can change those cells from differentiated, means adult cells, to baby cells. Maybe. It's a crazy idea, isn't it? It may work or may not. Anyway, by putting 24 genes, they found this kind of the color information, which is one character of the self-renewal and self-proliferation. Seems like it's working. And the next step, they tried, out of 24, they tried to take out one by one. Take out one. If it does not form this, that means that removed one is important or not. That's important, right? It's important, that's why if you remove it, then it does not have activity. By that method, they narrowed down to 4 genes. Using these 4 genes, if you overexpress of those 4 genes, they found that only 4 gene activations, and you can change this differentiated cell into embryonic stem cells. It sounds like crazy idea and unbelievable data, but actually shows that it's really working. And more important point is efficiency is quite high, and many groups tried to follow. Many of them succeeded. It's working, really working. There's some problems here though. For that gene expression, virus or lentivirus, those viruses insert those genes into the genome. Once you inject into the cell, those genes are introduced into the genome site, means it is remained in the body permanently, genetically modified cells. So that can be a problem because, genetically modified food, even food we don't want to eat. The cell is changing by putting extra genes into the body. That's big trouble potentially. And there is one of those 4 factors, one of them, c-MYC. This is oncogenes. Oncogene means which has a chance to make the cells grow fast and make the cells into cancer. And this kind of because of this kind of problem, whether this method can be used for this cell can be really used for human therapy. So this kind of induced pluripotent stem cell, in other terms, artificial embryonic stem cell, okay, originally differentiated cell, but induced iPS technically could be made. And then to reduce those dangerous potential, they tried to change this retrovirus into DNA virus. DNA virus at least, they do not insert those genes into the genome. So they just go into the cytosol, make a protein, and just work, and then later they can be removed. So maybe safer. And later even plasmid, just the DNA itself, and injected. Then after DNA function is done, then maybe removed. It's a similar way we take the vaccine, DNA vaccine or RNA vaccine, right? So DNA RNA injection, maybe the temporarily generated protein, and then they disappear, maybe safer. Another trial was, rather than those genes, why don't you put the protein? Make the protein outside of the body and inject those protein. Then protein cannot be replicated, so maybe even safer. And eventually, they tried to replace these genes with small molecules, like drug molecules. In the beginning, to replace those 4 factors one by one with the small molecules has been tried, and some of them was successful. If you look at those chemicals, drug molecules function, now you can see DNA methyltransferase inhibitor. Means they block DNA methylation. Some of them HDAC inhibitor, histone deacetylase inhibitor. So this implies that these molecules are all epigenetic modifiers. Original stem cell has open structure, and these compounds is remove those methylation pattern, add acetylation to the histone. Then the cells becomes from differentiated cell to younger baby cells, stem cells. So that is the reaction, and after some years, 2013, from Chinese group, they reported that only using 4 chemicals, no genes, only 4 chemicals, they can make embryonic stem cell, iPS. Well, this is quite a hot topic, but still efficiency is not super. So this was the hot paper, but chemically induced iPS. So still, there's a technically, they wanted to get a little more efficient method. And after that, there's another interesting paper, it's published in Nature, 2014. Hey, even you don't need compound. You just add 1 drop of vinegar. Vinegar and a little massage to the cells. Then you can generate the stem cell very efficiently, so-called the step cells. Unfortunately, this turned out to be another big scandal. The data was gained, manipulated. This happened in Japan, and this work is also didn't work. Eventually, there was a lot of tragic the things that happened. So far, up to here, this one does not work, but at least it seems like Yamanaka's factor works. iPS is real, and some of those factors can be replaced by chemical too. Then, current status. If you take any of your cells from your mouse, and then you can change those cells into your own stem cell. And using them, grow them, and ask some people in C5 building, they can make your nose. And if you want to have a little higher nose, then you can put your nose back. It's practically possible. That's a regenerative medicine. Okay, we're very close. So let's think about the relationship between stem cell and tumors. Stem cells, we said, it's genetically more activated. That's epigenetically we can modify. That's some basic things that we've done. Tumors, they grow very fast. And tumors, in many cases, also, they are quite active for gene expression. Many genes are activated. And then, in the video clip I gave you last week, there was the HDAC inhibitor as a tumor drug. Wait a second, HDAC inhibitor HDAC inhibitor, HDAC is histone deacetylase. Inhibit, then acetylation increase. We said tumor has a more gene expression. Now you make it even further gene express, then it's the opposite. If you want to treat the tumors, you may want to decrease the gene expression. That's the general direction, isn't it? But HDAC inhibitor is in clinical trial. How do you explain? Seems like this is the opposite. If you add HDAC inhibitor, you may generate maybe stem cell. But at the same time, you may generate tumors. So the biological system is not so clearly black and white. Seems like there's some genes. I said there's the genes helping tumor is called as oncogene. Right? Oncogene is overactivated, then the cells goes into tumors. Other type, opposite type of the genes, there are tumor suppressor genes. When these genes are activated, usually the tumor is suppressed. By strengthening those checkpoints, if any cells is going wrong, mutation accumulated, then they kill those cells. So these genes, once the cells goes into the tumor, oncogene will be shut down, the oncogene will be increased. Tumor suppressor gene will be decreased. Tumor is abnormal status. So either way, if you increase those gene expression, maybe tumor suppressor gene can be increased. Or if you decrease those genes, maybe oncogene could be suppressed. You may try either way, and then you choose those working ones. So in this case, HDAC inhibitor case, it seems like HDAC inhibitor increased the acetylation of this tumor suppressor gene. But sounds like quite dangerous. You need to check very carefully which one you touch. You know. Okay. So this is the relationship of those the tumor treatment using HDAC. So one more question here, given by you: cancer stem cells. Well, cancer stem cell and normal stem cells. Cancer stem cell, if you have a cancer stem cell, this cancer stem cell can make new tumors. Regular tumor, if you take and then inject into the different animal, it doesn't form tumor. But cancer stem cell, take a single cell, inject into a different body, it forms tumor. So this is like stem cell in normal status, but there's a big difference. So the question here is whether stem cell becomes cancer stem cell, or stem cell grows into differentiated cell and they become cancer first, and then this cancer, some part of the cancer becomes cancer stem cell. It's not very clear, but now we already learned that even differentiated cell, for a long time we believed cells are young and then grow and get old. It doesn't go back. That's what we have to believe. Right? But essentially, like differentiated cells, they have a chance to get younger by changing epigenetic modifier. Cancer stem cells, the cancer cell too, once they become cancers, majority of them is a differentiated cancers. Some of them, they may acquire the ability to become like like stem cells. So that is more reasonable to think that something is wrong. That wrong part is in the beginning epigenetic modification. Some part of the epigenetic modification, maybe smoke a lot, or maybe drink a lot, or under very bad conditions, and then epigenetic environmental effect change those cells. Once it goes over, now mutation happens. Once the mutation happens, the situation cannot be recovered. Epigenetic reactions, there is still chance that can be erased, can come back. Once the mutation happens, now they become cancers. If they die, then that's fine. If they do not die, they survive. And some of them, they require the epigenetic modification. They can become stem cell-like cells. But those cancer stem cells already mutated, that part cannot be deleted. Means cancer stem cell, there's no other way they become normal stem cells. then you need to select cancer stem cell and then kill them to cure the cancer completely. Okay, so today we have studied about the general flows of a stem cell and then moving those direction into differentiating stages in terms of epigenetic modifications, and then how to convert, how to revert these epigenetic markers to go back to the original state. So after here, this is the contents. Midterm exam will take the next week, Tuesday, same time here, 2 p.m. Okay? And I will the call. You may have a chance to make expected midterm exam problem. Then, if you want, you can put this up or send me by email by Saturday 9 p.m. If I like it, I will put it up onto the PLMS. Then your friends can look up to those problems too. That means among those problems, maybe you may see the same problem in your exam. Okay, so you study your materials. In this course, I will see the lecture summary from you, but this is one chance. You can make your own lecture summary or maybe some expected 예상 문제 currently for the midterm exam, then contribute to your friends. Select interesting problems, some thoughtful problems. Okay? Any question for this course and for the midterm exam?
The paper generation of fluid and stem cells is recombinant genes. They use small organic molecules and the range was like 1 micrometer to over millimole. And I think the concentrate of the organic molecule is really high, so
Too high?
yeah.
Okay.
Whether experiment is in vitro or they can apply over the in vivo too.
Well, let's just think about making the stem cells in vivo. How will you do? So you inject the molecule or you take those molecules, and how will you confirm whether which part is changing into stem cell? Maybe you can see ears growing, nose growing. So this is all in vitro. So you have the cells, most likely a skin cell culturing, and by adding these compounds, and some of them is yes, it's a super high concentration, millimolar concentration. But look at the structure. Structure-wise, this is very similar to vinegar. So it's not very complicated molecule like this. It's very simple molecules like metabolites. So that's why maybe high concentration of this maybe mild effect somehow maybe make the binding of histone to the DNA a little loose to make it a little more open is also possible. But complicated molecules, if you look at this kind of thing, this is mainly binding to specific target, and this is nanomolar concentrations. We learned the millimolar, micromolar, nanomolar. Okay? 1 micromolar is kind of a boundary. If it's higher than that, a little high concentration. Lower than that is kind of low concentration. So this is all in vitro test. You have differentiate the cell by adding these compounds. Check whether those cells are becoming stem cell or not. Okay, other question?
So, on genomic imprinting, I understand that as when you generate primordial germ cells, the epigenetic modifications get erased and then they somehow get put back on for sex-specific like imprints. And I read that the mechanism isn't that clear.
Maybe. And when they were in twin status, only one side is methylated, the other side is open, and this side is the active one. When they make sperm cells, so when they are divided into 2 cells, seems like this one, originally unmethylated one, there's a mechanism that to fully methylate during this stage. Same thing happens here. This one, when they make the egg cells, or for some reason this methylation pattern is removed for these other cells. So sperm cell and egg cell, they may have a special protein maybe, and then they recognize this sequence here, and then in here all methylated, in here all demethylated. Maybe that is the mechanism how they control all this. Just a speculation. Okay, then let's finish today's lecture here, and let's see next lecture.